Hello everybody, my name is Yenetzi Villagrana. Today I will tell you about my master research titled Prediction of Functional miRNAs from Single-Cell Transcriptomes. This work was done under the direction of Abreu-Goodger in La Mexico. MicroRNAs are short sequences of about 22 nucleotides that contribute to gene silencing. Through the years, many studies have demonstrated that microRNAs participate in tissue and disease development, as well as fundamental biological processes like apoptosis or cell division, so they are important gene regulators. However, we do not fully understand their role in individual cells. Fortunately, with the development of single-cell RNA sequencing, we can now produce single-cell transcriptomes that can help us study microRNAs involved in the regulation of different cell types. In this work, we use Motif Enrichment algorithms in order to discover functional microRNAs from transcriptomes of individual cells. This approach only needs gene expression information and the UTR sequences of these genes, so we can use this strategy to take advantage of public single-cell datasets to study microRNAs. The first thing we did was to download single-cell transcriptomes. In our case, we searched for datasets in the ABI single-cell expression atlas, and specifically we selected one called Tabula Moris, which consists of more than 50,000 cells of mus musculus from 20 tissues and 81 cell types. Using these transcriptomes, we made a differential gene expression analysis using HR, with which we generated a rank of genes from downregulated to upregulated for each cell type. Then, with this rank, we performed the Motif Enrichment analysis on the UTR sequences of the genes using Silamer. When we discover a significantly enriched motif corresponding to the binding site of a microRNA, we predict that this microRNA is functional in the corresponding cell type. We can also find other motifs that can be significantly enriched or deplete, but do not correspond to microRNAs. We use all these motifs as specific markers for each cell type, and we separate them into two types, the ones that are related to microRNA seeds and the ones that are not related with microRNAs. Next, we use the markers to see if we could predict cell types. For this, we selected 3 annotated test datasets from the EBI single-cell expression atlas, and we performed the same process described before: differential gene expression analysis, motif enrichment analysis with Silamer, and the definition of markers per cell type. We then use hypergeometric tests to define the maximum enrichment of markers between each of the known cells of the test datasets versus each set of cell type markers from the Tabula Moris dataset. With this logic, the Tabula Moris cell type that shared most markers with the unknown cells was determined as the identity of these cells. Finally, we compare our predicted identities with the annotated identities and determine the percentage of correctly identified cells. On to my results, here I'm showing you some examples of the curves of enrichment that we obtained with Silamer. In the x-axis, we have the sorted UTRs from downregulated to upregulated, and in the y-axis we have the significance of the enrichment or depletion. With Silamer, we could predict several functional microRNAs that we expected for certain cell types. For example, here I'm showing you a plot for neurons and the enrichment of the set of microRNA 124, which is an important cerebral regulator. On the other hand, we also found novel cell-type microRNA associations, for example microRNA 9 with the Berman glial cells, a microRNA that until now was only reported as an important regulator of cerebral tissues and neurons, and also microRNA 30, which is related to cancer of the kidney and lung, but we found it consistently in endothelial cells. Concerning the performance of our markers, overall we observed a better performance using the markers that are not related with microRNAs than with the markers related to them. For a dataset composed of cerebral cells, we correctly identified all cells. However, for other cell types, identification was not as good. For the immune cells, the identified cell types were in general related to the annotated cell type, but the intestinal cells were the most problematic. We attributed this to the similarity between cell type, as we see using TSNIDs, not shown. So, in conclusion, our results highlight the usefulness of Silamer for the prediction of functional microRNAs for individual cells and the discovery of novel microRNA cell type associations. We suggest that specific profiles of motifs that are not related to microRNAs can help distinguish cell types. We hypothesized that they could be motifs related to RNA-binding proteins. We propose these markers as a complementary tool to improve existing cell identification and classification techniques. Some relevant bibliography, and that's all. Thank you very much.